Hey, and we are back. Jonathan and Radka, are you guys there still? Can anyone hear me on the line?
We're here.
Okay,
We're here.
perfect. So we have you up next to talk about applying genetic insights to ash management. If you want to share your screen again, if you have the screen that you wanted to.
Sure. We'll share it. But I think before I share it, how we envision this is really much more, of a discussion, and we have some questions that we put out there first on that document that we shared, but that we also have on some slides, but they're by no means meant to be exhaustive or to constrain the discussion. They're just examples of some things that we were thinking about. And we think this is like an amazing group to run this stuff by and get ideas. And the very first thing that we were about to open that presentation, but the very first thing is actually something that Zane discussed in a recent presentation for tree species in peril. But I don't think, unless we had a bit of a problem here where we lost the stream for a while. We didn't hear him discuss it today. So we kind of assumed he was, and that everyone would be familiar with it. So I don't know, Zane, did you discuss the self-incompatibility system in FNIGRA today? We didn't catch it if you did.
I mentioned it briefly, so I think everyone in the room is aware that we have fun cryptic self-incompatibility and stuff going on.
Okay. Okay, great. Because so if there's anything you want to add to it, this is kind of like, I guess the first thing that popped into our head was when you were talking about the self-incompatibility system in FNIGRA. So the question we had is, how does this affect management in the following specific way? That there are all these initiatives. I think they're spearheaded first in New Hampshire, and then other states and other cooperators have adopted them to go out and inject small numbers of trees of probably all the native ash species, but in particular F. nigra, to quote protect in situ diversity. But what we were wondering, both Radka and I, after we saw your presentation on the self-incompatibility system in this species, is how would that affect the number and distribution of those trees to be protected in situ to retain that genetic diversity? And then kind of flowed from that a whole bunch of other questions that we had in terms of, taking the insights as far as self-incompatibility, just mating systems more generally, cryptic species and all that, and how can we translate this into any guidance for managers and for management, because a huge percentage of our audience and our interactions is giving guidance to managers in terms of what they can do to enable long-term conservation and restoration of ash. So are there, can we develop any particular guidelines based on any of these insights you and the other geneticists and taxonomists have developed? But we can, I guess, start with this as an example. So anybody's thoughts? Zane, anybody else out there?
I'm zipping over to the microphone. Sorry, Jonathan. So yeah, this is a great question, great point of discussion. And I think there's, can give a little bit more background and also give some nuance to some of this. So the first thing is that we did identify the presence of the previously described self-incompatibility locus in black ash. We've also seen it in blue ash and in some of the species from Mexico. However, so the way that we identified that was essentially comparative with the other Eurasian species that have been molecularly validated and physically validated with cross-pollen assays, because this self-incompatibility system is ancestral in Oleaceae. And so this is largely us saying, we found the same gene, it is intact, it has the same structure, same number of nucleotides or of amino acids in the protein. And so we don't see any reason why it shouldn't be functional, but I do also want to just nuance that by saying we have not tested and validated that in our North American ashes. We see no reason that it shouldn't be functional, but just throwing that out there. Those groups have also already developed primers. So we talked really briefly earlier about barcoding, for example, for species diversity. We're not there yet, but in terms of self-incompatibility assays to identify incompatibility groups, that exists. Those primers are online and freely available. People could order those and PCR out those genes today. It's presence absence. So that already exists and is possible. I think there's probably some more agile ways we could try and implement that for things like black ash management. But in terms of maintaining in situ diversity, this is a great question. Jill Hamilton's group, along with Natalie Isabel and the Canadian Forest Service and many others, APCAW, have been doing a lot of work in essentially collecting across the range of black ash and range wide from some of these other species as well. So I think in the near future, they may be better positioned to try and answer some of these questions genetically than we are. But there's still just a lot of unknowns and a lot more data that needs to be generated to really understand how self-incompatibility is distributed on the landscape and within stands. Like, is it a 50-50 ratio or is it not? Because there's a distinct possibility that you have an imbalance between compatibility groups. It's just something we don't know.
Can I have how many loci and how many alleles?
One loci, two alleles. That was going to be my comment.
Yes.
I think we actually have a pretty, if we're going to have self-incompatibility, this is kind of the best case scenario because it's diallelic. There's only two. So it's kind of similar to being male or female. So if you have a big collection of black ash or a big collection of blue ash, you're probably going to capture both compatibility groups. And it's a hemizygous locus, which means you either have two presents or one, and it's not, there's no reason for us to suspect it's not reasonably close to 50-50 on the landscape. It may not be, but there's no reason to think it isn't.
Right.
It's not like some of these really complex self-incompatibilities that have tons and tons of groups. So that's the good news. I guess I just wanted to point that out. There is some good news.
Absolutely not. I completely agree with that. I just wanted to throw it out there that we're still at we have not proven this in the North American landscape, but there is a really nice amount of literature coming out of Europe to support that this should function the same way. We don't see any reason that it would not.
Right.
So.
And it doesn't seem to be functional in our dioecious species. So it's not like we've got self-incompatibility. I mean, you wouldn't really need that if you're dioecious. So it's not, it's only in a worry for Excelsior is a weird one. So they have kind of a whole weird thing going on. We don't have an Excelsior type polygamy system in the US, or in North America. So that's good news for us, I think. Well, that we know of, maybe we'll find one.
Excelsior is insect pollinated also. Does that help out?
So Mary, I don't know if you guys can hear Mary okay, but she commented on the different pollination mechanisms and whether Fraxinus excelsior is insect pollinated. I don't personally recall. Most of the ashes are wind pollinated, but some are definitely insect pollinated. I don't recall exactly which ones. Alan, do you know?
No, I'm not sure. It doesn't have a corolla.
Okay.
But
I think all the North American species make them pollinated.
not when you get out west or something. Petals, yeah. Oh, yeah. In the Southwest, there are some that have petals and likely are insect pollinated. Eastern U.S., I think they all are wind pollinated.
So I think our concern with these like in situ conservation efforts is that, they're typically quite a small number of trees that they're just because of cost and logistics involved, that they're injecting, quote, protecting to begin with. And I don't know really what guides the number of trees. I think there may be some attempt to be protecting both sexes, and I'm putting that with quotations around it, and that I think generally, the emphasis has actually been on overwhelmingly protecting female trees because they're seen as seed sources. And so that then raises another question, right? If you just have a bunch of females and one male, then, that's raises a couple of issues. And including if you also want to have not only multiple males, but multiple males of a particular compatibility type, and the same thing for females and so forth. So we're just wondering if there's any guidance that can be developed, because I think the people who are doing this would act on it. They want this to make sense. They don't want it to be a dead end.
You want to go ahead, Kathleen?
Yeah, I agree. I think having more guidance about how compatibility groups might inform those kinds of in situ efforts would be really helpful. Did I understand right that you said that you don't expect the self-incompatibility to be a factor for dioecious species, or?
Right. Well, they're naturally self-incompatibility because they're either male or female flowers. So the
Right.
self-incompatibility locus has actually just like disappeared. It's fallen apart in
Okay,
those species. So it's only the hermaphroditic species that we think have the self-incompatibility.
so only the hermaphroditic species would
Yeah,
would
so
have
only,
that issue. Okay,
From what, like black and blue, ash. Everything in the Melanoides is dioecious. It's just that the self-incompatibility is interesting because it's cryptic, right? At least with the dioecious species, once they flower, you know what you're working with. With the self-incompatibility, they have perfect flowers, and you just can't tell if your crosses are going to work or not until you know the compatibility groups. You can't see it morphologically.
Well, there's also probably some additional nuance to throw into this as well. So a lot of the Eurasian species, in section Ornis in particular, are androdioecious. So they have males and hermaphrodites. I'm no joke that Fraxinus has almost all of the sex systems and all of the things. So without getting too much into the weeds, we know that the self-incompatibility locus is propagated primarily, but not completely, through males in section Ornis. However, it appears that in some of the other sections that may not be the case. And so if you want to talk about, if anyone wants to chat later about all the nuances, we've got a paper coming out soon talking about some of the origins of dioece and a couple of sections of Fraxinus and the different mechanisms and things that go into that. So happy to talk about more about that, but it would be probably too lengthy of a conversation.
And it is really interesting in thinking about crossing Manchurian and Nigra, because Nigra is a hermaphrodite with self-incompatibility and Manchurian is dioecious. How's that going to work? So we know they can produce fertile F1s. What happens after that? I think there's going to be, for crossing, that's going to be quite a weird thing to work out for some of these species.
I know Dave wants the.
Oh yeah, Dave's on. We keep asking him, what do those flowers look like, Dave?
It's a hot mess.
Fraxus Manchurian hybrids are a hot mess. So since Dave's not jumping on, I'll just say we have been doing the F1 crosses between Manchurian and Nigra in both directions. We have the hybrids that were developed at the Morden Research Center in Canada. The Northern Gem, Northern Treasure were widely planted as street trees for sort of the north, like Minnesota, colder regions. And there's another third of the F1. So we have our F1s, we have their F1s. We've made back crosses to both parent species, and the crossing is really confusing. And I think the SI locus, when we ever have time to sit and really sort through it a little more carefully, is going to help us understand why some crosses didn't work. But crosses do work. The F1 is fertile back to both species. We have some that worked both directions back. And we just, that's a side burner project. Again, we only have so much capacity and we focused it in the We focused principally on the green and the white, and we sort of backed away a little bit. I mean, we have a little still interest in it, but we've principally backed away from the hybrid breeding model. So there is crossing data though that can be sorted through to kind of sift through that, and that's probably where, I mean, that's where you're going to be able to sort it out. Then if no one's going to pursue Manchurian by Niagara crossing, it's going to be within Niagara problem. And if that comes down to the two compatibility groups layered in with the weird gendering and black ash. But if it's two groups, you should, I think your point, Meg, that you should be able to capture it just as readily as you capture males and females in green and white ash. Hopefully it's not as big of a management or like planning criteria that way.
You don't have reason to suspect it's going to be a huge imbalance of one compatibility group to the other. So if you had, we can do the stats on if you get 10 of these into an XC2 situation, what's the likelihood you capture both groups? Probably pretty good.
And just to chime in and sort of take it back to Jonathan's original question for how do we do this in a tractable way on the landscape? The barrier to entry for identifying what compatibility groups you have in natural stands of black ash is reasonably low. What brass tacks, what you actually need is someone who feels confident they can extract DNA. It doesn't even have to be great DNA because it's just one locus. Beyond that, you need primers and a PCR machine. And to be able to stick it on a gel and say, cool, we collected 10 individuals from this population or this stand, and you throw that PCR product on a gel. If you get a band, it has the locus. If you don't, it doesn't. And so it's reasonably tractable to do that, and for different groups to get involved with just sort of basic, reasonably common molecular lab supplies. And, if anyone wants to talk about that, what that might look like, we're super happy to chat about how to do that.
So I guess one approach would be to do that. And I'm just wondering, for a lot of the, managers who are out there, they probably don't have any of those facilities. So one approach would be to partner with, research institutions that would have that, or more advanced, maybe breeding centers have access to that. And I guess the other approach would just be to go big enough in terms of how many, how many trees you're protecting, that you just should have the odds on your side. I think a lot of these efforts, though, are really very small numbers of trees. And so I think that's where our concern lies, that, they're very small numbers, and they also tend to be heavily, skewed towards females, so that you may have that, that one male or the two males, and, what are the chances of that? So I'd like to investigate, I think Radka and I would like to investigate, both of these approaches of, coming up with some guidance in terms of numbers, if there is kind of a shotgun blast that's going to be big enough to capture everything that's needed to be captured, and or, partnering with facilities that could do that testing. And I think it also then ties back into the broader question of what guidance, well-informed guidance is needed regarding the number and distribution of trees to protect in situ for maintaining genetic diversity. There may be some literature behind the numbers that they use, or it may just be, a practical sense of this is how many we can afford to protect. But it would be, I think, nice to investigate, developing some guidance and a kind of scientific basis that takes all these factors into account, so that folks aren't, wasting resources on something that's not going to really accomplish anything.
So as an example, for Vermont, state is injecting or protecting their black ash populations, at least one in each county, and they were injecting about 12 trees per population, and it was 2/3 of females versus whatever the rest of the trees that they ended up to the number of 12.
This is Kathleen. I think some of the numbers that they're using are based on the paper that Charlie Flower and I published. So I'll be presenting that later today when we talk about tree treatment and associational protection. So you'll get a little more nuance on that. But in doing that paper, that was a paper with white ash, and we were not considering self-incompatibility issues. But we did find that, after you've treated 10 trees to get the biggest bang for your buck for diversity, the best thing to do is to move on to another population and treat another 10 trees rather than treating more trees in that population. So they bumped it up to 12 just to give themselves a little more of a buffer. And I don't know how they made the decision to have more females than males in that population. But I believe that's what it's being based on, and you'll see more information on that later this afternoon.
Sounds great. Really happy to hear that. And
I do just want to, sorry, Jonathan, I was just going to say, I do just want to plug really quickly as well that a lot of the range-wide collections, population genetics type work that has the people who have the appropriate data to start trying to answer these kinds of questions are Jill Hamilton's group from Pennsylvania State University and her student, Kira Lopiccolo. They've done a lot of range-wide collections of black ash, and this is in collaboration with the Canadian Forest Service, et cetera. Green, they've done green, white. They have a lot of information on Oregon ash as well as velvet ash from the Southwest that's currently unpublished. So in terms of thinking about local genetic diversity and things like, what is the local effective population size that we have across the range in different little pockets, they really have the right kind of data to start informing the basic ways that we should try and structure this management. So just throwing that out there, that they may have more to say and more information on that.
that sounds great. They need to be pulled into this.
They are Julie's on.
Oh, they're there. Okay, they're there. They are in
Yeah.
this. That's terrific. And I'll just move on to the next page. And again, these were just a brainstorming session that we had, and I don't know, they may, I tried to strip it down a little so it's not too repetitive, but so you kind of all these similar questions. Should a sufficient number of functionally male individuals be chemically protected? Again, there's this focus on females because of their seed bearing capacity, but do you need to get enough males so you don't have this kind of bottleneck through one male or whatnot? And then more generally, is there adequate guidance? And this is something I'm really looking forward to hearing Lane's presentation. And again, not to beat this point to death, so should it take in system the species, and it sounds like it should. And then we spent some time on the mating systems, and anybody who has anything else to add, I'd love it. We're really learning from you. But the next thing also that we're learning from the genomics is all those cryptic species, the not just widely known species, right? But we have all those infraspecific taxa, varieties within F. pennsylvanica, and individual species often lumped by non-taxonomists and so forth. So I think it would be really useful. I think we think it would be useful to the extent. Now, a lot of these things are so cryptic that you really cannot distinguish them in the field, but some of them maybe have some, signs the more closely we look. And even beyond that, if you are doing some kind of a molecular analysis to identify trees in the field, how can we integrate that to make sure that some species don't fall through the cracks in terms of, be it lingering ash detection and material collection or seed collection or whatnot. It would be really good to furnish whatever tools we can to managers and to the broader audience that's involved in these on the landscape efforts, out in the field, to take into account these species that are largely unrecognized. So any thoughts on that?
Yeah, to your third point in particular, about targeting different species. I mean, I do think that there's significant value in that. Just so, for example, the case of white ash and Biltmore ash. So white ash, again, diploid, Biltmore ash is a hexaploid. They're both part of the same quote unquote species complex, same lineage. But from some of our data, we've found that Biltmore ash appears to be largely a lowland species. It grows sympatrically with white ash much of the time, but white ash also has a much larger or a much broader extent up to higher elevations. Maybe that's a thing, maybe that is a drought tolerance thing, just with availability of water. These are things that we don't know. So for a lot of these species,
Oh.
Echo. We don't have great ecological profiles for a lot of these species. And just given the amount of cryptic diversity, I would maybe even contend that we don't have a great ecological profile for Fraxinus pennsylvanica and Americana, considering how wide-ranging they are and how much diversity throughout that range that we just don't understand. So I think all of this needs a really good holistic revisit from people with a much broader background than I have, folks like Kathleen in particular, people who are community ecologists, et cetera. But just from the species diversity standpoint, I do think it's important, due to the different ecological profiles, to make the best attempt that we can to capture these species, and also not to ignore the hybrids. I'll give you one example. The Northern Research Station's breeding program, they have a small handful of admixed individuals, which I think Mary might touch on later. And we even found one individual that appears like it's a one-to-one, just like an F1 first generation hybrid of green and white ash. So they do occur, and some of them are lingering. So there is potential there or utility there to try and conserve some of those taxa. Again, this really gets more into the weeds about, what are the different taxonomic units that you're actually trying to conserve. I think we just embrace the gradient and we capture as much as we can.
Yeah, I think there's so much enthusiasm for this group to find ash, but it's difficult even for experts to know what they're looking at. So for the people who are doing the taxonomy and who are doing the research and getting it done, the more you can provide keys, written morphological details, and trainings, I think is going to be really helpful for people who want to do conservation. Because right now it's just tough for us to do it, any of us to do it effectively. So those could be products, I guess, would be really nice. And should probably be part of a longer term research plan to sort out a lot of the cryptic stuff going on.
Yes. Yeah, I have been starting to gather images and so on. I think Julian's been doing the same thing. And also we're, thanks to Zane and to flow cytometry, we're finally getting a solid idea of what we need to be looking at and what things we don't have to be looking at. But yeah, it is important. Absolutely, you don't just ignore the hybridization, but at the same time, it's probably important whenever you're working to have an idea whether you're looking at a rare viable hybrid between two groups that cross very often, or a happy looking hybrid that's got an unbalanced genome because it's a diploid and a hexaploid crossing. You probably want to know, the more you know about when you're looking at something like that, you just need to think about them differently. And absolutely, we need to get better identification aids and also probably find somebody who can put it all up online so that it's easily accessible.
Hey, can you guys hear me? This is Dave Carey. Hello?
Yep, we can hear you.
Yeah.
Okay, cool. I've been driving, so I haven't been able to chime in yet. Two quick things. With respect to the Niagara and what you might be saving on the male side, I think the math is pretty simple. You need at least 6 males to get like 90% chance of having both incompatibilities in a stand. So if you want immediate advice, my recommendation would be save the equal amount of males and females and not imbalance it because of this incompatibility piece. if you're only going to save 12, do 6 and 6. That's just my suggestion. Other thing I like to share is that we are currently taking photos of everything in our program to try and help with this sort of keying things out, since the genomics are being done with them as well. So we're trying to develop a database of the winter stem morphology and leaf, and scoring the papillae and that sort of thing as well. So we're starting that on our end.
Hey, Dave, while we have you, in the chat, Natalie Isabel had asked Mary, have you checked the F1 with genetic markers? And she just told me that I should ask you that.
Checked it for.
Oh, well, the one F1 hybrid, oh, I'm sorry, which species?
Manchurian Niagara.
Here, say it in there.
Manchurian Niagara.
Manchurian Niagara, have we tested? Yes. Actually, Laura Kelly did that while we were working on an Excelsior project. So yes, we do have confirmation that we have F1s and backcross were both verified molecularly.
And I'll just jump in too and say that Meg and I briefly spoke with Laura Kelly last week as well. She's indicated that to us that she's willing to share some of that data also. And we're also whole genome resequencing both the parents and all of the reciprocal progeny for that Nigra and Manchurica offspring group to try and sort some of that out. But I do also just want to nuance that when it comes to self-incompatibility and sex in Fraxinus, what we have seen in the other taxa is that for the most part, it is likely that self-incompatibility likely segregates with sex. So you are probably not looking at self-incompatible or males with the locus, males without the locus, females with the locus, females without the locus. You're probably looking at like males don't have it, females do have it, and that's what's driving that maleness, femaleness type of thing. We don't know that for sure, but you probably do have two effective sexes, is the guess.
As far as we know.
Except that Nigra changes over its lifetime and within the canopy.
That's a whole nother,
It
yeah.
can. So,
Well, we have a genome for one of those trees, so maybe it's about time we do a deep
Excelsior has the self-compatibility and the sub-dioece, but we thought we didn't have that in North America, but I guess I don't know anything about
Just to go back to the management question, whether or not it's like that or not, at the worst case, you're talking about two loci, one for gender, one for sex incompatibility, both with two alleles at an expected frequency of 50%. So, you've got your little Punnett square thing, you've got about 25% in each box, right? Is the expectation. Those aren't the alleles that are going to be hard to capture. The loci where you've got 20 alleles and you've got 3 dominant alleles and you've got 17 really rare alleles that happen at most maybe a half percent each. That probably is happening in ASH, especially in some of these polyploids. Those are the ones that we're gonna lose. And capturing an adequate number of rare alleles is probably where we want to focus our intellectual effort at this management question, maybe more so than these common alleles. I mean, we're going to get them for free, functionally. That's just one thought on that.
I think the concern was not that we will, lose these alleles globally or regionally, more that if you're trying to maintain a persistent, population there and you're just maintaining a few, and they happen in the numbers so small, and you're not structuring things to make it likely that you have both of these alleles, that you'll then, effectively prevent sexual reproduction between them. But did that work or not?
Yeah.
So I think this has been really productive. I just want to get to the last few questions, and this did not mean to be, again, exhaustive. So it seems to have, the discussion is spurred a lot of answers and a lot of further questions, and feel free, to share them. So I think like one thing we've been focusing on for the most part is the in situ conservation, but then there's also guiding collection of material for resistance breeding, to take this into account, and which is probably more along the lines of what you were just speaking about, and also more to also, and maybe this is something, I don't know, Mary, that you're going to address later, but incorporating these genomic insights in terms of mating systems and taxonomy in terms of designing breeding plantings. So, and to an extent, these all complement each other. And for instance, if you're trying to maintain in situ diversity by injecting a certain number of black ash, then you're taking those black ash out of the potential pool of lingering ash, because once they're injected, unless you had already previously identified them as lingering ash, those are then excluded from the pool because, hey, they're maintaining their health because they've been injected. So these all kind of need to be, integrated. And there are, we have partners, for instance, who are trying, all of the above, who are doing seed collection and want to also enable lingering ash detection, and also inject some trees for in situ conservation, to the extent that that doesn't inhibit the ability to find lingering trees. So, I think that these, broadly called genomic insights, which are really fascinating to us, can impact management approaches and conservation approaches, I think, in multiple ways. And I guess I'll put my emphasis on management, because even if you're just collecting material for resistance breeding and doing that breeding, at another site, still, when those managers are coming up with a plan in terms of, we'll treat some trees and we'll cut some trees and we'll collect seeds from trees and whatnot, that it's good to, factor all these considerations in from the start, to the extent that it's necessary and relevant and practical. So anybody else's thoughts on anything? We have a few more minutes. We're willing to hear them, and any other questions we should pursue. And we really, again, a lot of our interactions on a day-to-day basis are with managers, and managers who are seeking, guidance in terms of what they can do to best achieve conservation and restoration. So we're really committed to working with folks from this group and beyond to try and develop guidance materials that would be helpful.
Yep, we're going over to Mary, or sorry, Kathleen.
Adding one further complication to the, if you treat the trees, then you can't determine whether those would have been lingering ash or not. We're also, there's evidence from a number of studies, including ours, that untreated trees that are near treated trees also receive some degree of protection, and we don't know at what distance that phenomenon operates. So it also makes it hard to, even if it's an untreated tree, if it's very close to a bunch of treated trees, it may be alive just because it's near those treated trees and not because it's truly a lingering ash. So that's another puzzle that we're needing to get more data on to figure out.
Yeah, so I would add that we're certainly aware of that, and it's actually something that we've been wrestling with when managers are asking us, even right now, for guidance, and that it's not just the individual treated tree, but potential, effects, broader, more associational effects and whatnot. So that's something that we definitely need to keep in mind, and we'll leave it at that. So yeah, absolutely.
So I actually have a question about that, because I've heard this too, and do you know the mechanism behind it, whether it's lower pest pressure or like systemics through rootgrass?
Skip that slide because it's a mess. So we were talking about that this afternoon. We don't know yet what the mechanism is. We're doing analyses now. We've ruled out one that we thought was the mechanism, but we are looking at others and trying to figure out. So it may, the one we ruled out was that it would work like herd immunity in people where, you know, if you have enough people vaccinated, then the unvaccinated people are protected. It does not have a relationship with density of protected trees in that manner. In fact, it looks like the opposite relationship. You'll see that this afternoon. So that, scratch that one. The other possibility that we're working on, trying to figure out if we can analyze, is if we created lethal trap trees accidentally, if some of our insecticide treated trees are in fact stressed trees that were ash canopy 3 and 4, and so they're kind of drawing in emerald ash borer. The lethal trap trees was a strategy used in kind of the SLAM slow ash mortality techniques. So that is a thing. So that could be part of why we see that. We're also looking at whether there's some kind of below ground translocation of insecticide, either through leaf litter or root exudates or root grafting. So we're looking at, on a fine scale, actual proximity of trees to see if, you know, are we mostly seeing this effect in trees that are right next to each other, in which case that would point towards something like that. So we're to be determined, but we're trying to figure out what the mechanism might be.
Kathleen, just to your comment about more data on this, we may have some like light data that we could potentially share, but we also, Great Smoky Mountains National Park is doing a lot of like trunk injections of imamectin benzoate specifically in clustered areas versus not. So we do have a lot of those trees, whole genome sequenced, identified to species, all that. But we also, they're all cataloged in TreeSnap. So we have a good amount of morphological data, and then surrounding sorts of what do the trees look like around? Are there other treated trees around, or are there not? It sounds like you would need a little bit higher fidelity information than what we have, but the Park Service also might be willing to share some of that information. So maybe we could get you all connected and try and, yeah.
Hey, Kathleen. Hey, this is Dave Carey again. Can you hear me?
Yes, we can hear you, but if you need Kathleen, she is grabbing some water right now.
Oh, okay.
All right.
You know what? I will wait till her talk later and ask this question then.
Okay, that worked out because we're about 2 minutes away from starting our next session. So this could be a great natural segue. Unless anyone has some very quick,
I can talk about Built Moriana if you're interested.
Can you do it in like 1.5 minutes?
Yeah, I got a really quick anecdote about Built Moriana, which we have been neglecting, unfortunately. I recently met a retired director of forestry school in the UK. He said that the forestry industry in Kentucky has been very confused, as we have. One of the reasons they have not been harvesting white ash from Kentucky for baseball bats is they go to Pennsylvania and New York, where they know it's all Americana. There's no Built Moriana. Because it turns out Built Moriana has been confusing people in Kentucky. Sometimes they call it upland green ash, which is not. And it usually grows in the hills of Kentucky on slightly poorer, less fertile, more stressed soils. Anyway, but if you go back to, sorry, wrap it up now. 1982, there was a paper done by Armstrong where they looked at the fiber length of wood from the hexaploid, and it was longer in the hexaploid than they didn't call it Biltmore. So I'm wondering still if, you know, Biltmore could be a pretty good baseball bat species, but no one's ever really tried it because they think it's green. So that's my anecdote.
Thank you for that, Julian. Yeah, if there's still more questions related to all of this, well, I guess Jonathan and Radka, they are kind of tied up for the rest of the day, but we'll be keeping track of them in the chat. They also have this document, it's on the Padlet if people want to write comments on it. And will you guys be able to join us tomorrow at all, Jonathan?
So we kind of shuffled our schedule, so we should actually be available this afternoon again. So we can't join you for lunch, but